Section B, the cell cycle. Okay, now let's get the details on how your cells divide. It's called the cell cycle.
Let's say we have a cell that wants to replicate and divide. First, it duplicates all its cytoplasmic contents, and there's an order to that. The ribosomes and organelles are duplicated in a stage called the GAP1, or just G1 phase. Next, the cell duplicates all its DNA in a stage called synthesis, abbreviated as just the S phase. Then the cell checks to make sure everything is duplicated, which is called the G2 phase. Finally, the cell divides, called the M phase, or mitosis.
Let's get into more detail about the S phase, or DNA replication. This is the process by which the cell makes a complete copy of its entire DNA code, so that when it divides into two cells, each new cell has identical copies of the code.
Remember we told you before that the beauty of those weak hydrogen bonds in DNA is that they allow the DNA to unzip and replicate. So to replicate DNA, you unzip a segment so you've got two separate strands. Then the new strand is formed off the template of the old strand. This new and old strand now stay together and form one of the new copies. In this system, each new copy has one old strand and one new strand. This method is called semi-conservative replication, because one strand of the old helix is conserved in the new helix. Now we need to add just one more thing to this explanation of DNA replication. Earlier we said that the bases A and T are attracted to each other, and G and C are attracted to each other. And this is how base pairings and replication can occur. But there are little helper molecules that bring the complementary bases together. You can think of them as a matchmaking service.
Yeah, this is Chuck Schick here on Base Connection, Public Access Channel 34. This is a game show here where we pair up bases, and I'm sitting here with my good pal, Adenine.
Yes.
How you doing, Adenine?
Yes.
Well, Adenine, let's see who you have to choose from. First, there's guanine, and there's, of course, cytosine, and then we move on down the line to thymine. Who are you going to choose?
I choose thymine. Chuck.
Okay, well, before we hear about your pairing, let's hear what the audience had to say about it. Ah, look at this. The audience chose thymine. You know what this means, Adenine?
No.
It means that you get a paid trip right here by the public access station 34 to unzip the DNA double helix.
When DNA replicates, there are special proteins that serve to unzip the helix and actually make semi-conservative replication occur as described. The most important of these proteins is DNA polymerase. There are a few different types of DNA polymerases, and each does a slightly different task.
After it synthesizes all of its DNA, the cell goes through another gap phase called G2. Now, one of the reasons for the G2 phase is to check to make sure everything's in proper order and all the necessary parts are replicated before the final process of division. This actual division is called mitosis, and it's abbreviated as M. All the phases except for M are collectively called interphase. So, G1, S, and G2 are together called interphase, and M is just called M. The time it takes for a cell to go through each of the four phases depends on the type of cell it is.
We are about to begin mitosis. Set interphasers to stun.
One final note. Most cells don't just duplicate and divide forever. At some point they stop during one of these phases and remain in that phase the rest of their life.
So to recap, we asked, what about growing bigger? And we answered that the way to grow bigger is to make more cells. And cells are made in a duplicate and divide manner. Here are the steps again.
1. The cell duplicates its cytoplasmic organelles, called the G1 phase. 2. The cell duplicates its DNA, called the S phase. 3. The cell checks to make sure everything is duplicated, called the G2 phase. And 4. The cell divides, called the M phase or mitosis. This is known as the cell cycle, and it's a carefully controlled process.
Now you know that the cell goes through the cell cycle, which consists of 4 phases: G1, S, G2, and M. Let's talk a little more about mitosis, or the last phase.
To watch more free educational videos, visit our website, studioforlearning.tv. Stretch your brain online.